ready? We can get started here. Welcome to another breakfast with WACh. It's great. How many have we had now, Grace? Is this about 10 or so? Maybe more. This is great. So thanks everyone for coming. This is really exciting for me and for our group particularly, I think, because our speaker, who you can hear about in a second, Dr. Anna Weil, is coming from Mass General now, but she's en route to Seattle, where she's moving to make a home and be part of our Division of Infectious Disease starting, I now understand, July 1st.
Yes.
She brings some really interesting skills and experience that I think are really complementary to a lot of the work that we're doing in the center here. And so I'm really excited to hear her talk and for all of you to sort of start thinking about sort of what collaborative opportunities might exist here. So I think one of the areas that really struck me when meeting with her before and reading through her materials is she really brings a really interesting breadth of knowledge from field experience in Malawi, Ethiopia, and Bangladesh to really sort of basic science mechanistic pathway work in the lab, particularly focused on diarrheal disease and the role of the microbiome in modulating pathogen interactions with the gut and also with the immune system. And a lot of the work that we're doing from the sort of classical epi side in diarrheal pathogen work is really focused on sort of defining these mechanisms from an epi perspective, but we really have lacked sort of the expertise in the more sort of basic science and basic immunology, pathophysiology of disease. And so I think I'm really excited to have you coming, and I think you will find yourself a sounding board for lots of ideas, like it or not, in the future. But I'm really excited to hear her talk, and yeah, I'll let you jump in right from there.
Thank you. Thank you for the very kind introduction. I'm excited to meet all of you and learn about the work that's being done here and find out, how we can work together and how I can contribute to the really important problems that this group is working on. So my talk, I'll just talk about a couple of different areas of how the gut microbiome impacts Vibrio cholera infection. And first, just to give a little background, Vibrio cholera is the bacteria that causes cholera, which is a severe diarrheal disease that causes millions of cases every year. It's tough to estimate exactly the burden of disease, but much of that burden is in children under 5 years old, especially the mortality data. This is really unfortunate because the vaccine candidates that are currently being increasingly used in cholera really don't protect children well, especially young children. They don't have the same durability of protection, and the amount of protection they gain is much less than adults. Also, from an epidemiologic standpoint, the severity and frequency of cholera epidemics has been changing over the last 10 to 15 years for many reasons. One of them is that the strains that emerged in Haiti and in other areas since 2010 do produce more cholera toxin and are actually more virulent than previous strains that circulated. Global travel, as I'm sure you are aware, in Haiti is well proven cause of the epidemic there, and this has affected other areas as well. In addition, the rising ocean surface temperatures in many areas seems to also have an impact, since this is where Vibrio cholera has an environmental reservoir in warm ocean water. So currently cholera is endemic in 50 countries. Most of these are in Southeast Asia and Sub-Saharan Africa. And endemic meaning there's sporadic cases that occur all year round. But there are also epidemics, which are a little more visible in the news. But most of the cases still occur in endemic regions. So I just wanted to go through quickly, clinically, what cholera looks like. I don't know if you guys can see that. Can you see that at all? A little bit, maybe? Sorry, it's kind of a dark picture. So this is a patient who presented to the International Center for Diarrheal Disease Research in Bangladesh. She has many of the features of a person presenting with severe dehydration. Her skin texture doesn't look quite right. She has sunken eyes and depressed mental status. And patients like her present this way. They're healthy adults typically who present after 8 to 12 hours of vomiting and diarrhea. So the mainstay of treatment in this setting is rehydration for patients like this who can't drink. Oral rehydration solution. The treatment is with intravenous fluids. So the fluids that are lost for a patient like this look like this. This is called rice water stool. It looks like water after rice has been cooked in it, so it looks quite different than a lot of other diarrhea. And in patients with severe cholera like this, they can have up to 1 liter of output of this rice water stool per hour. And after people improve from their rehydration, they can then start drinking oral rehydration solution, which is really the mainstay of therapy for moderate disease, and even patients with severe disease after they begin getting rehydrated. And in 12 hours, they tend to look actually quite well. And at the ICDDRB in Bangladesh, patients with cholera who don't have comorbidities like malnutrition or pneumonia are typically hospitalized for under 24 hours. So you can see it's kind of a dramatic presentation that's very severe and acute, but actually gets better very rapidly. And in places that have the infrastructure to care for patients correctly and the technical expertise, the case fatality rate is actually very low. Antibiotics are also used for treatment, but these are not, they're not required to save lives in the setting of cholera. Antibiotics really just are used in order to decrease the period of symptoms and, importantly, reduce the need for hospital resources when people present with disease, since, as you know, people who have these severe diarrheal diseases typically are in places that are low resource. Thank you for the lights help. So today I'm going to go through a few different topics. First, I'll talk about the changes in the gut microbiome during and after cholera. Second, the relationship between the gut microbiome and susceptibility to Vibrio cholera. And along with that, what we've learned from in vitro experiments about how the pathogen interacts with gut microbes. And then we'll switch gears a little bit to talk about oral cholera vaccines and protective immune responses and how the gut microbiome may impact these areas. So before I jump into that, I just wanted to also show this map, which emphasizes that the Ganges River Delta in the area of eastern India and Bangladesh, where all of the rivers kind of flow down from the Himalayas and drain into the Bay of Bengal. This is the ancestral home of endemic cholera, where it has circulated for hundreds of years. This is a map of all of the waves of the 7th cholera pandemic, which has been ongoing since the 1960s. And all of the patients I talk about, all of the cohorts that I work with, and all of the samples I'm going to discuss today are from this area. And they're all from the ICDDRB in Bangladesh. Although we also work with many cohorts from Haiti, today I'll just be focusing on the ones in Bangladesh. So first, to talk about bacterial changes during cholera, we'll be talking about patients like this. These are patients at the ICDDRB in Bangladesh being treated for acute cholera. So we essentially assess their gut microbiome at the time they present with cholera and then follow them for 30 days afterwards. So in assessing their gut microbiome, I'll just kind of first show a summary slide of those results. Just have a pointer. Can you guys see that? Can you see that? No, it's only on my screen. I'll just gesture in that direction. Oh, that's a good idea. I can do that. So, so this is a principal coordinates analysis plot, which is a commonly used plot in gut microbiome research, but they're not always very intuitive, so I just want to explain a little bit more about what you're looking at. So you can think of each data point as all the phylogenetic information about a person that's compressed into one point, and this kind of exists in a three-dimensional space. But what is shown when you see these figures is just the rotation of that image until you see the most resolution between the groups you're looking at. So the important point of that is to say the closer together the dots are, the more common and more alike those bacterial communities are, and the further apart they are, the more different they are. So this is in healthy controls with the purple circles, and you can see that on day 0, when people are presenting to the hospital, their gut microbiome has already shifted considerably. And this is due to the infection itself, the efflux of fluids they have from the diarrhea. And then by day 7, or I should say by day 1, when they're all the way over on the right, now people have also received antibiotics, which has further obliterated their gut microbiome and a bunch of oral rehydration solution, typically. But you can see that by day 7, they're already starting to move back over to where the healthy controls are. And by day 30, they're making their way almost there. So from this work, and in addition to this, a functional analysis of what the bacteria in these groups were doing at these different time points, we concluded in this study that changes in the stool gut microbiome after cholera recover by day 30. So to get into a little more detail about what the bacterial groups are in this progression. This is a plot showing some groupings of bacteria that we created to explain this succession. So on day 0, with the red bars, you can see that many people's microbiome is dominated by cholera. And then quickly, they begin to have these, what we called early stage organisms, which are present on day 0 and then also on day 1. So these are the bacteria that show up and sort of take over at the time when there's this blank slate after all these things have happened to the gut. And these are bacteria that really live in the environment or in the hospital or are present in people's mouths. So these are just kind of the first things to show up, and they're able to proliferate at that time. By day 7, we start to see some facultative anaerobes and then obligate anaerobes that are beginning to emerge back into this space, and people start to look more like healthy contacts, which are all the way over on the left. So using this information as well as the functional genetic information, we created a gut microbial succession model after cholera. So these are cartoons of the gut lumen all the way over on the left. At baseline, we know that there's this diversity of organisms with a competition for different resources that's ongoing. And this is supported both by oxygen that diffuses from the blood flow around the intestines that's diffusing into the lumen, and then also by carbohydrates, which are produced in the mucus by epithelial cells and also are from people's diets. So at baseline, the microbiome is dominated by the large blue anaerobe and a few little aerobes in yellow there. So with infection, all of these commensals are killed or washed away, and the oxygen starts to build up that's diffusing in from the bloodstream. And that high level of oxygen and the carbohydrates that are building up, since there's no bacteria to eat them, those two factors really support this bloom of the aerobes that we first saw when people have their microbiome beginning to get repopulated. In this setting, the oxygen tension is quite high, and anaerobes are not able to survive very well. But over time, the oxygen tension is lowered by all of the aerobes that are using up the oxygen, and the anaerobes are then able to start repopulating this space. And then by day 30, we see that this balance is more or less reestablished, with many different species taking up these niches.
And did all these patients receive antibiotics?
Yes, all of them did receive antibiotics. I think if we had people who didn't receive antibiotics, which is a study that I think could be done, but probably ethically in the gray area, I think we would see something similar, just because from studies of the duodenal microbiome where Vibrio infection occurs, there's just like almost nothing there on day 2. I'm not sure antibiotics they may further impact them, but it's hard to say for sure. Any other questions about that part before I go on? Okay, so next I'm going to move to talk about household contacts of cholera patients. So these are the people coming and going from the hospital, visiting their family members. And we know from historical studies that this group does have a high risk of infection. And several years ago, when I was in Bangladesh as a medical student, we did a study of clinical outcomes in household contacts to get a bit of a more contemporary look at this problem. So this is a study of 1,000 household contacts of cholera patients. They are enrolled in the study at the time their household case presents to the hospital, and then we ask them questions about what happened during the week prior, and then we follow them prospectively to see if they're shedding bacteria and to see if they're having symptoms. So as you can see from this, there's kind of a good bimodal epi curve of what's happening in these households, and from this work we found that 20 to 25% in later cohorts of household contacts do become infected. So they don't all have symptoms, and actually very few of them develop dehydration, but learning about this high burden of disease in this group gave us an opportunity to enroll some groups of them that had no signs of infection at the time of enrollment in the study, but who then went on to become infected usually in the previous or in the next few days. So we're able to get pre-disease samples in a subgroup of people and compare those to people who were uninfected during the entire study. So this is a study design of many of the results I'm going to be talking about more. So using this study design, our group and others have identified several clinical factors predictive of susceptibility to disease. First, low vibriocidal titer. This is a serologic measure of exposure to cholera. It's not a perfect measure, but it does measure exposure, and this is an important factor. And independent of this, young age and blood group O status also impacts susceptibility. But overall, these factors really don't predict infection very well, and it seems that there's a large portion of susceptibility that's still unexplained. So our group has spent several years trying to understand more about what's in this gap. It does appear that there are some innate immunity genes that impact susceptibility. Not surprisingly, people who live with others who shed bacteria, especially for long periods of time, are more likely to become infected. Malnutrition is a risk factor for a longer duration of bacterial shedding, although it's unclear if this impacts transmission within the household. So when I became an ID fellow, I was really interested in asking about the gut microbiome, which was an area I hadn't studied at all before that, and understanding how susceptibility might be impacted. So this is a subgroup study for that question, and this is using the same study design where we enrolled household contacts after case presentation. At that time, we were able to assess their infection status and then follow along and see if some of them developed infection in this subgroup. There were 22 who became infected and 54 who remained uninfected. All of them were uninfected at the time of enrollment. So we assessed their status regarding infection, but also profiled their gut microbiome at baseline and during follow-up periods. So we collaborated with a group at Duke University to create a machine learning model to identify predictors of infection in the gut microbiome. So we split these samples into two groups. The first was used as a training set, so we used their disease outcome, which we knew, and their gut microbial factors, and then used supervised learning in a feature selection model. We identified gut bacterial predictors. So then we applied those gut bacterial predictors to the testing set and combined this with the clinical factors we know impact infection. And from this generated a statistical prediction, or really a probability of how well that model predicted the disease outcome. So for these results, I'm just going to show you two area under the curve models. So these are, again, really a measure of the probability of how well the model predicts the outcome based on whatever the inputs are for the model. So on the left side is the holdout validation of what I was describing with the training and testing set. The clinical model, you can see in the green, doesn't really predict the outcome much better than chance, which we were expecting. But the microbiota model actually does much better just with the gut microbial information alone. And when we combine those two, we actually don't find much of an improvement, which indicates that there's a lot of overlap or redundancy between what we're measuring in the clinical model and the microbiota model. Something like age, which is linked to the microbiome and linked to susceptibility, is one possible explanation for this. And then on the right side is a follow-up model to this that we use to understand how many microbial taxa were important to measure for this prediction. So we again used the testing and training sets, but we trained the model on 90% of the samples and then applied it to 10% and reiterated that 300 times with a random assignment of those groups for those household contacts. And we found again that the microbiota model did perform better with not much improvement in the combined model. But when we added, there were 4,100 bacterial taxa in this model, and the total species of the gut microbiome from these participants. We found that you really only need around 100 to make this prediction. So this is where we got the number for the top 100 bacterial taxa that predict Vibrio cholera susceptibility. So these are the groups of bacteria that have been a focus of ongoing work regarding interactions with the pathogen and interactions with the host immune responses. So this is a plot that highlights some of those groups. There's a few things I want to just mention about it without going into great detail in all these species. So this is a slant plot showing you the relative abundance of these bacteria on the left in the protected group, and then the decrease in this case in the infected group, and the same thing with kind of weaker relationships in those that were in the groups we found that were infection associated. So at first glance, you can see there's some bacterial species that are on both sides of this graph, and that some of them recur several times, such as Prevotella copri. And the numbers that you see are these are the operational taxonomic unit names of the genetic group of bacteria. So this is an arbitrary number that's just assigned to this group. But what it indicates is that this group of Prevotella copri is genetically distinct from this one or this one that might have the same species identification. And this highlights a problem in the microbiome and microbiology fields of, we have these taxonomic identifications that have been created over many centuries and decades, and now that we have genetic information about these bacteria, they actually might not be that related at all. And Prevotella is the genus that's a great example of this, because they have up to 50% genetically distinct sections, even in the exact same species. So this highlights a greater problem in the microbiome field, which is that identifying bacteria to the species level is sometimes not adequate to understand about what the functionality of those bacteria are, or to learn really about what's happening with those bacterial groups.
So, quick question.
Yeah.
I might have missed how you identified the associations on this slide and the prior slide. What tests were you using?
To identify the...
Which species to target the top
Yes.
100? I guess that's how you did it.
Well, we identified them first with 16S, our V4 sequencing that's the area of the ribosome that you sequence to identify the taxonomic group, which is where these kind of numbers of operational taxonomic units and levels of relatedness are identified. And the way we identified the top 100 was really by putting all of the gut microbes and the clinical outcomes into a model that then basically selected which bacteria were linked to those clinical outcomes.
Like a regression or lasso or something?
We started out with that and didn't find very much, and so we moved on to machine learning with random forests and a few other models, which I can talk more about, to make that correlation. Again, it is a correlation, what we're seeing with these clinical outcomes and their relationship with these bacteria. Does that answer your question?
It does.
Okay.
One other question.
Yeah.
Just with these infection-associated bacteria, you see a lot of the strep species and other things. Which
Yeah.
You've obviously alluded to the fact that these may be signals of early recovery of the microbiome. Do you think that this somehow reflects that the infection risk is associated with a recovering microbiome, be it from malnutrition or from antibiotic exposure
Yeah.
or disease?
It's a great question. So the streptococcal group has been identified and related bacteria in a previous study that we did as a group that is present in early recovery, as well as in people who seem to be more susceptible in a smaller group. That's been shown by others as well. I think that in this setting, what it indicates to me is that these are really markers of an unhealthy microbiome. These are markers that the typical anaerobes maybe aren't present in the numbers they should be. I mean, strep is part of the usual gut microbiome, but really shouldn't be dominant in this setting. And also, you allude to the next slide, which is what is really the relationship between these microbes and these clinical outcomes? Could these just be a marker for some other factor, like enteric enteropathy or antibiotic use or something related to the diet? And that's definitely a possibility. Even if those things are true, it will take time to tease out what's cause and effect and what's really impacting susceptibility. But it's certainly possible that these are simply a marker for something else. It could also be that these bacteria affect pathogenesis or host immune responses through other means, including regulation of inflammation or host immune responses, or that there's a niche that's being taken up by these bacteria that kind of excludes Vibrio cholera at that moment it arrives in the gut, and that there is some, what we would say, colonization resistance that is conferred by certain groups that really doesn't allow Vibrio cholera to colonize effectively due to resources that it is not able to have. It could be there are metabolites that these bacteria are making that somehow impact Vibrio cholera as well. Did I answer your question totally? Okay. So these are the areas that kind of when I step back from the study and think about what's really happening here, these are things that I'm thinking about, and to further investigate these questions, we started by isolating these bacteria from this population, from these household contacts, which took some time, but we have made some progress on that and are now working with these in the lab to understand more about these relationships. So just to show you one example of this, that was our first observation. This bacteria called Paracoccus aminovorians, which is a little known soil aerobe and not a very common member of the gut microbiome. We noticed that during cholera, when everything else had been washed away, this bacteria seemed to be flourishing, and it wasn't clear why that was happening. So we began culturing in these plots. The first set of initials is the bacteria that's growing, and the second set is the bacterial supernatant, or the metabolites from that bacteria that were growing that initial bacteria in. So when we grow Vibrio cholera in Paracoccus, around Paracoccus metabolites, we see there's much more growth than in controls, although not as much as in fresh media. We would, of course, expect much more growth in fresh media. So from this, we started to look further into these other bacteria to find out if they were impacting the pathogen itself. So how is it that metabolites, such as what's in the supernatant from these bacteria, can impact susceptibility? So I've been spending a lot of time thinking about what the microenvironment is in the small intestine when Vibrio cholera is introduced. So first, it has to survive through the stomach and then get into the small intestine. And when it arrives there, it has to make its way swimming through the outer mucus, which is filled with bacteria, the inner mucus, and then also onto the epithelial cell to colonize and make cholera toxin. So there's a few ways that the microbiome could impact this path. And one of them is influencing nutrient availability. Also, some bacteria make molecules that alter metabolic functions of pathogens. And there's also the possibility of disruption of interspecies signaling, which in Vibrio cholera is very important for causing disease. So all these things are happening, when a person is first exposed, and are harder to study in people who already have active disease, but this is really the area that I've been focusing on the most. So to demonstrate some of the testing we've done, this is a picture of kind of the genes that control virulence factors in Vibrio cholera. And the first place we looked was at biofilm formation. The formation of biofilm in the gut is an important factor for Vibrio cholera to sort of start congregating in large amounts when infection begins. And we actually found no relationship between biofilm formation and the top 100 bacteria that we are working with. So next we looked at the two most important virulence factors that the pathogen makes, both of which are needed for causing disease. So the toxin co-regulated pilus is the mechanism by which the organism attaches to the epithelium, and cholera toxin is what causes the watery diarrhea. So we looked at the production of mRNAs for cholera toxin and the toxin co-regulated pilus, and the results were the same, so I'm just showing the cholera toxin results. These genes are controlled by similar upstream regulators, so it wasn't surprising that they were the same. But what we found was that when we cultured these bacteria in the supernatant from bacteria from our top 100, there was up to 100-fold increase in cholera toxin production, specifically with the Paracoccus again, and a few other species. So it's not clear exactly what the mechanism is of this change, but 100-fold increase, or even this 10-fold increase, is a huge impact on the clinical state that a person might have with the microbiome present. So this really emphasized to us that the microbial species present at the time of infection could have a huge impact on whether or not Vibrio cholera grows well, and whether or not the infection is mild, moderate, or severe.
So you grew up each of these bacterial species on just a plate, right? They weren't exposed to a host. So it's just pure bacterial supernatant.
Yes, anaerobically. And then we have also now applied them to an organoid model of duodenal epithelium, which is hard to do anaerobically, but we're doing it aerobically first. So kind of moving ahead to look at other models of how we can understand these relationships. And I'll show some pictures of that a little later. So then going back to our virulence factors, the next thing we looked at was quorum sensing signals. And quorum sensing is critical for Vibrio cholera's ability to cause disease. What this means is that when the bacteria is present in when there's very few of them around, they signal to each other with small molecules. There's a few different ones, but the one we're going to talk about is called autoinducer 2. So when they're present in small amounts, they don't put out this small molecule, which tells the other bacteria that are around that they should colonize the epithelium, produce cholera toxin, and stay there and grow for a while. But when they're present in a dense amount, like such as in a biofilm or in a florid infection, they produce AI2, which then tells them that they should leave because they're going to run out of food. So they stop making cholera toxin, they stop colonizing, and they actually regrow their flagellum, and they leave the host to go find another host. So if quorum sensing is changed, which I will explain how it is, then that would also impact how infection progresses. So other people have previously noted that specific gut microbial species, such as Ruminococcus obium, which was on our top 100 list, impact Vibrio cholera quorum sensing by producing AI2, which turns on the quorum sensing receptor genes in Vibrio cholera, causing a decrease in toxin co-regulated pilus and cholera toxin. So we wanted to look into this in some of our other species to find out if this was also happening. And we've tested, this has 4 of the organisms, but we've tested around 11 now, and they all produce AI2. So there seem to be multiple mechanisms by which the gut microbiome is actually making a big impact. So here we used Vibrio harvii, which is another Vibrio species that luminesces in the presence of AI2. So this is a measure of the optical density of the bacterial culture of Vibrio harvii growing in the presence of the supernatant from these bacteria, including the Ruminococcus obium from the previous study. And so far, we've found that this is potentially also an important factor. So I'm going to switch gears to talk about immune responses. Yes, that was a great question. We do have a group of control bacteria that were found in the gut microbiome of the people in the study, but were not ranked in, actually we used the top 500 as a cutoff for selecting controls. I didn't include those here, but we do keep those as well. Some of them also make AI2, consequently. So we have a lot more to figure out about what exactly is happening here with these relationships. So I'm going to switch gears to talk about oral cholera vaccines and immunity. So this is an area that I worked in previously before I got interested in the gut microbiome, and I'm going to just keep it brief because I want to make sure we have time to talk about other things. So just as background, immunity to Vibrio cholera infection is durable after natural infection. This has been studied most in people who live in endemic areas who probably receive kind of constant low-level exposure, like boosters. So it's not clear exactly how long this lasts. For example, in North American healthy controls, it's only been tested up to 3 years where people did have immunity. We think that protective immunity most likely operates at the surface of the intestinal mucosa. Since Vibrio cholera doesn't penetrate into the epithelium and it's a very non-invasive organism, it seems most likely that there's some type of secretory IgA or some other effect that is mediating this protection. But these questions are not solved, even though people have been studying cholera for 100 years. So this is still a work in progress. To measure immunity and to also measure immune responses to vaccination, the serum vibriocidal antibody is most commonly used. There's no threshold level of this antibody that indicates full protection, but it does indicate exposure and increases in age. So in endemic areas, people have a detectable vibriocidal antibody usually by their early teen years. This antibody is directed against the O-specific polysaccharide of the LPS, which coats the tail or the flagellum of Vibrio cholera. So oral cholera vaccines have been around for at least 50 years, and they haven't worked very well until recently, but they are increasingly effective. And since the Haiti outbreak, there's been much more political will in using them as part of cholera control. The current vaccine that is most commonly used, that's part of the WHO stockpile, is a whole cell killed formalin inactivated organism with no components of cholera toxin. So there's so much enthusiasm for the new vaccines and increase in political will that the World Health Organization recently set a goal of reducing cholera by 90% by 2030, which is coming up pretty soon. The problem with vaccines is that they, as I mentioned earlier, are not equally effective in all populations, especially in young children. They also elicit different levels of protection in endemic areas versus non-endemic areas for reasons that are not well understood. And one big difference between children and adults and people in endemic areas and non-endemic areas is the gut microbiome. So that's one reason I'm interested in finding out what the impact is of the gut microbiome on vaccination. So to study this, I used a cohort of vaccinees in Bangladesh, and I first wanted to understand if giving the vaccine, which is an oral vaccine that's absorbed in the small intestine, if giving the vaccine alone changed the gut microbiome before we try to get into how the microbiome is impacting immunity. And we found that it doesn't, and this is also true in polio, oral polio virus vaccine and rotavirus, that there doesn't seem to be an impact on the microbiome itself from the vaccine. So this is another PCA plot, and you can just see from your eye that these dots are not grouping in any way. So this is, again, a 30,000-foot view of people before vaccination and after, showing that there's really not a difference, at least on the community level, of the gut microbiome after vaccination. So next we looked at people who seroconverted by vibriocidal titer earlier versus later, which is important in epidemic areas where the disease spreads rapidly and vaccine administration can take time. So a quick seroconversion is something that is important. So in this heat map, these columns are each one participant, and they're split into faster versus slower seroconversion. And it doesn't look like coloration doesn't look perfect, but there's much more dark blue in this faster seroconversion area. So this, at least in the first row, which is the Ruminococciaceae group. So this is a family of bacteria that is well known in autoimmune disease to cause immunomodulation, mostly due to short-chain fatty acid production. And we found this, I'm using this result sort of as an example, but there are others from this study that indicate not just short-chain fatty acid production, but also the amount of toxigenic LPS present in someone's microbiome at the time of vaccination do seem to impact their vaccine responses. So my real interest in this study was to find a way to identify with a little more focus which bacterial groups we should be studying to understand these differences. So the way that I decided to do this was by identifying IgA positive coded stool bacteria. And these are the bacteria that are being recognized by the innate immune system and that the mucosal immune system is reacting to. And we study these by first staining the stool with anti-human IgA and then sorting the fractions using a magnetic column and then enriching them further using flow cytometry. So we can really divide them into stool bacteria that are IgA coated and those that are not. And using this method with our pilot group of samples, we found that there's an increase in IgA coating of bacteria after vaccination, which is not surprising since you're introducing an antigen and stimulating the mucosal immune system. And using sequencing to further elucidate what's going on in these groups, we found that they're quite different and that groups such as Enterobacteriaceae, which is contains kind of your E. coli and other groups that are more inflammatory to the immune system and have toxigenic LPS are IgA coated, along with some other groups. And the goal of this work, which is actually just ongoing this week, is first using shotgun metagenomics to understand what the function is of these IgA positive organisms compared to the rest, and then also correlating these IgA positive organisms with immune responses to vaccination. And this is really so that we can identify what bacteria specifically or what bacterial groups or what genes in which bacterial groups could be important for stimulating the innate immune system at the mucosal surface during vaccination. And my goal is to study this further in children and adults so that we can pinpoint if there's something we can do with vaccination in children, like giving an adjuvant probiotic with vaccination that might further stimulate the immune system at that time point. So with that, I'll just talk for a couple minutes about ongoing work in my group, which I will be bringing here. First, that we're working with new models that I mentioned earlier to study these top 100 predictive bacteria at the level of the pathogen itself and also at the host level to study immune responses. And we're using currently a duodenal organoid model, which is a kind of a single cell layer that we grow of these cells that is derived from we start with really a biopsy of the human duodenum and grow these, put them in these transwell cultures on the left, which then allows you to measure the electrical impulses of this layer so you can assess how healthy it is. And these are some EM pictures of one of those layers done by my postdoc Elena. So you can see that there's like the outline of the specific cells, and then in between there's the little dots which show the microvilli. So this model has been working pretty well for us for the last year. This is a picture of after one of the models after 4 hours of Vibrio cholera infections. You can see it's not looking quite as good. It has some cracks that are going along the tight junctions and not looking as intact as the first picture. Along those tight junctions, you can see organisms often that are kind of hiding in there and also penetrating the epithelial cells, or colonizing them, I should say. And on a close-up of the epithelial surface, we can see not just you can't see my arrow, the biofilm that's produced that's kind of keeping all these organisms together, and then also these long strings, which we think are the toxin co-regulated pilus that are then coming out of the organisms and colonizing the epithelial surface. So our hope is to use this model further to study not only the host responses from the epithelium, but also integrate macrophages into this model so we can understand at least the beginning of what is happening in the innate responses when we apply the supernatant and metabolites from these bacteria to the model, as well as studying the virulence factor production from the organism itself. So secondly, I also alluded to earlier my interest in studying the duodenal microbiome. In many microbiome studies, obviously the stool is is the easiest sample to get, but it's really a measure of the luminal bacteria in the rectum and in the colon. And as I mentioned, Vibrio cholera infects the small intestine, which is quite distal, and this is also where oral cholera vaccines are absorbed and where colonization occurs. So it's really this much more upstream area that we should be most interested in. And there are a group of patients in Bangladesh who have had biopsies of their duodenum during and after cholera. And in these people, this is another type of microbiome plot that just quickly summarizes not just the diversity of organisms, but also the abundance. So it takes into account both. And you can see that on day 2, there's barely any bacteria and there are not very many of them. But that again, by day 30, the microbiome is looks as if at least at this high view has been reconstituted. So I have ongoing sample collection in Bangladesh to get at these questions of really what's happening in the duodenum, what organisms are there, and what's happening to them during infection. And then lastly, as I didn't talk about in detail, but I alluded to, and I can expand on if you guys are interested, how can we understand why it is that children don't respond to vaccines the same as adults? Is it the microbiome? Is it their immature T cell subsets, particularly the T follicular helper cells, that don't allow them to generate the same memory B cell responses that we think really mediate long-term protection? And this is also an area of ongoing work. So with that, I just want to say that all of this work is a group effort. And I want to thank my mentors at MGH, Jason Harris, Ed Ryan, and Steve Calderwood, those in my lab, Meiti, who does the flow cytometry, and Elena, my postdoc, and our collaborative group in Bangladesh, as well as individuals at the Broad who are working on the vaccine microbiome study with me, and for the machine learning model at Duke, the laboratory of Lawrence David. And that's it. I look forward to answering your questions.